Hey, it's Mr. YS, and this is a clip on DNA structure and RNA structure. So here are the questions. What are nucleic acids? What are the two types? What are nucleotides? And how can a nucleotide be represented in a simple diagram? Okay, so DNA is deoxyribose nucleic acid. And then making up the D and the N and the A, so DNA. And then if we're thinking about what RNA is, the other type, it's ribose nucleic acid, so that would make RNA. But when we write it out, we remove the last bit, so we get rid of the S and the E, and it's just deoxyribonucleic acid, or ribonucleic acid. And these are both polymers because they are made up of monomers and they are large biological molecules. And so the monomer that we're going to have a look at for these is called a nucleotide. So it's these deoxyribonucleic acid and ribonucleic acid are both made up of nucleotides. But the nucleotides are very slightly different because in deoxy, as we'll see later, we change the structure. We have removed oxygen from the ring structure. So we're going to learn how to draw a nucleotide, and we simplify it a lot. So here we go. I'm going to rub and reveal this. Here is our nucleotide. So the first thing is that we draw a circle. So one of our first key bits is a circle, and then we're going to join it to a pentagon. And you can see that we've joined it in a particular spot, and that is important for the diagram. And then we join that to a rectangle. So I want you to draw along and try and label it. So the circle is representing phosphate. It's a phosphate group, PO4 3-. So it's got this negative charge to it. And that's important for separating DNA. We can attract it, it'll attract two positives because of the phosphate. Around here we have, this is either, depending on whether it's DNA or RNA, and we're going to look at DNA structure today, this would be then deoxyribose. So this is deoxyribose, which is, because it's got 5 in the ring, it's a pentose. It's a pentose sugar. So deoxyribose, a pentose sugar, or it could be ribose if we were talking about ribose nucleic acid, RNA. Over here, this rectangle then, this is the bit that represents the nitrogenous, because it contains nitrogen, it's a nitrogenous base. It's got nitrogen in there, and we'll see that. So we need nitrogen in the diet to make DNA and RNA, and also to make protein. Okay, so we've labelled our phosphate, our pentose sugar, and our nitrogenous base. And these could be one of the 4 that we represent with the letters A, G, C, T. And these actually have different shapes. They're not very much like a rectangle, but they do bridge a gap. They're a bit like the rungs of a ladder, especially when we join it together into a double-stranded structure. Okay, so this bit here, we can see that this side, this bit, we're going to see it's going to connect up. This is the sugar phosphate backbone. And then these are the bits, the nitrogenous base is like the rungs of the ladder. So around over here, can you identify what these different things are? What's the first one? What's the second? What's the third? The first one here, because we've got PO4, it's our phosphate. Down here we have our sugar, our pentose sugar, And then over there we have our base, our nitrogenous base. Now you can see around here that we're going to get into labeling. And so to the right of, it kind of looks like a house. I think of them as being, you know, it's like a house. If that's the roof of the house, we look up to the roof and we can see that it's this kind of the right top of the house is labeled as carbon 1. And then we go around carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and then it goes to the pentose. So this is going to be important, knowing which the carbon is. So going from the right hand side and just counting round up to carbon 5. Because the fifth member of this ring group is actually the oxygen, which is there. So here's our quick summary. We have nucleic acids are the two types of polymer. They are found in the nucleus. Well, DNA is always found in the nucleus. RNA leaves the nucleus and it goes to the cytoplasm. But they are certainly produced in the nucleus, so they're largely found in the nucleus. So the two types are DNA and RNA, and they are nucleic acids. They're a polymer formed from the monomer. The monomer that we just drew up there was a nucleotide. So a nucleotide is those three things linked together. One nucleotide is all of that. It's the phosphate, it's the sugar, and it's the base, the nitrogenous base. So that's the monomer, and it's going to repeat again and again. So nucleotides, we can say that they make up polynucleotides, but we call that polynucleotide. Most of the time we just call it DNA. We don't really talk about polynucleotides very much. Okay, so we talked about the three parts. 5 carbon sugar called a pentose. Up there the negatively charged phosphate group. And a base which contains nitrogen because it's a nitrogenous base. So our next question down there is, how do the nucleotides in one strand, how they link together? So we're thinking in just one strand at the moment. Well, in one strand, if we draw some repeating units of these, we have one nucleotide there. And now we need to link together another nucleotide. And so to link them together, we form a covalent bond. So all of these lines, as we are drawing them as a solid line, it means it's a covalent bond. We're going to start drawing some dashed lines or dotted lines, which are going to link together two sides, but these are not covalent bonds. They're going to be hydrogen bonds, which are much weaker. So these are strong. They're strong covalent bonds, and the covalent bonds are running down the backbone. So this means that as we keep linking it, the whole backbone is very strong. So if we heat up DNA, we won't be breaking these. Even if we heat it, the covalent bonds remain. Okay, so this is why we call it a sugar-phosphate backbone, because we're going phosphate to sugar to phosphate to sugar, and we're using covalent bonds between these carbons. So here's our quick summary. How a nucleotide is linked together. It's through covalent bonds form between the phosphate of one nucleotide and the pentose sugar of the next nucleotide. And they are between, if we label over here, it was 1, 2, 3. 1, 2, 3. So this was 4, and then although we're not drawing it, it's kind of a line, it's carbon 5 up there. So if we're thinking that it's carbon 5, we can see that it's between 3, then there's a phosphate, then there's a 5. So if we're looking at the direction of this strand, we can see it's 5 there. It's running 5 all the way down to 3. And we call this 5' to 3'. We'll come back to that in a minute. So this overall creates a strong sugar phosphate backbone, alternating sugar and phosphate groups, with the bases linked to each sugar. Again, that is all of these bonds that we've looked at are covalent bonds. So what are the differences between DNA and RNA? And so down here we have 2 pictures. See if you can spot the difference. What's the difference between those? And which one is the sugar that makes up deoxyribose nucleic acid, and which one is the one that makes up ribose nucleic acid, and why? So we can see down at the bottom, here we have 2 oxygens. We've got OH and OH as part of those hydroxyl group. A hydroxyl group is an OH group. Whereas up at the top, we have removed, if we're thinking about going between that one and that one, there's the removal of oxygen. So this is the deoxyribose. And this is ribose. So the first difference between deoxyribose and ribose is that one, and ribose nucleic acid, between DNA and RNA, is that one contains deoxyribose and one contains ribose. But we also have some other differences. So in DNA, we have the bases adenine, we have guanine, we have thymine, and we have cytosine. So what's the difference with RNA? And how can we tell it apart just from looking at the bases? Well, we have got something slightly different. We've got adenine, guanine, cytosine, they're all the same. We get AGC, but now we have a different, we do not get, it gets replaced, thymine is replaced with uracil. Down to our sugars that we talked about just then, one RNA has ribose with that extra oxygen down there and deoxyribose with the oxygen removed. Number of strands, well, it's a single strand and it's often in a linear shape, but not always. So it can form some weird shapes. It can form a T shape, and we're going to see something called tRNA. But it makes up often, so the linear one that we will see is called mRNA, messenger RNA, but you can get this kind of, it's kind of a T-shaped thing, which is tRNA, and we'll see that later. But it's always single-stranded. For DNA, it is 2, and we're going to use this term antiparallel. It's an antiparallel because one, it's like the road system. Our road system, we have one road running one way and one road running the other way, but you always drive on the left-hand side. And so that's the same for DNA. We've got 2 opposite directions that we could look at. And it forms this double-stranded helix because it turns. Alright, so we're going to try and draw a full diagram of DNA. And so when it's talking about drawing a full diagram, we are thinking of drawing both of the strands. So we're going to have these two strands, which we said were running in opposite directions. They are anti-parallel. They don't just run parallel, that would be both of them running in the same direction. They run anti-parallel because one runs in the other direction. So here we go, we're going to try and draw circle down to our pentagon, out to our rectangle, and then we're going to label those 1, 2, 3, 4, 5. And the linkage was between 3. We go 3, then we do another circle, and then we go down, and we do another house, another one of these pentagons. Okay, so we can label all of these things. This is our phosphate. This is a covalent bond to our pentose sugar, which was deoxyribose. Then to our nitrogenous base. I haven't left myself a lot of room there. You're going to see in a moment what I'm going to do. Then we need to draw the rest of it. And so the easiest way to do this is to turn your page upside down. Now, pretty hard for me to do. Can I get all of that? Can I turn it round? No, I can't. So that makes it quite hard for me. So on your page, you should be able to turn it round. If I turn it round, it's just going to rotate the screen. If you turn it round, it should be easy for you to draw. So I'm just going to go and stand the other side. And then what I'm going to do is, to make sure they match up, I'm going to start at the rectangle. And then I'm going to draw the same thing that we had on the other side. I'm going to link it to my pentagon. I'm going to draw a phosphate up at the top. I'm then going to go down to a phosphate. I'm going to draw my next house. Draw out. Here's where they don't quite match. That's why it's easier to do it. Yeah, when you turn the paper. And so now I've got two strands drawn and they can link. So they're anti-parallel. I've drawn them together. Now the final thing that we would want to add to this is the names of some of the bases. So adenine, A pairs with T. And guanine pairs with cytosine. C pairs with G. And we can remember CG3, they all rhyme. A and T, T for 2. And we can see up here that we have got 3 lines which are linking G and C. There are 3 hydrogen bonds, so we can label these as well. These are hydrogen bonds. And there are 3 hydrogen bonds between cytosine and guanine, and only 2 between adenine and thymine. Okay, what else can we label about this? We can also take a box and do dashed lines to highlight what our nucleotide is. So we can say that this is our nucleotide. So we talked about the phosphate, the pentasugar deoxyribose, the nitrogenous base. We've labelled them and showed how they go together. We've shown the different different numbers of hydrogen bonds, and we've also shown that it is an anti-parallel strand and that it is double stranded. And here's a nice diagram showing the same thing. So we can see that we've got adenine and thymine always going together, and cytosine and guanine. So why is that? Well, it's to do with the size of, if we're thinking that this is a ladder and these are the rungs of the ladder, it's to do with the sizes of those. So you can see that A and T are different sizes. And adenine and guanine, they are larger. These two, I remember it as pure green apples. Pure green apples, I don't know why, some student came up with it. And that reminds me that adenine and guanine, pure for purines. So there's 2 different types. These 2, and you can see that the purines are the larger rung of the ladder. They take up kind of 2, whereas thymine and cytosine, they're the smaller rung of the ladder. These are the pyrimidines. So the pyrimidines are smaller. And if we try and bond these together, because of the positions of these groups on them, only adenine and thymine will be able to bond to each other, and only guanine and cytosine will be able to hydrogen bond because of the position of these things that are involved in hydrogen bonding. Okay, so here's our quick recap. DNA is made up of 2 anti-parallel strands held together by hydrogen bonding. Each strand is made up of a chain of nucleotides which are linked by covalent bonds, we said, between the phosphate and the sugar. It forms a double helix, and we get hydrogen bonding between adenine and thymine or cytosine and guanine. Now here you cannot just say A to T. That is not a mark. They're quite picky about that. And the way that they bond, A bonding to T, AT2, CG3, the way that they bond is called complementary base pairing. And we're going to see that term used a lot. Complementary base pairing. Because their shapes and structures complement each other so they can form hydrogen bonds. Okay, final part. How did Watson and Crick use models to discover the structure of DNA, to work out that it was anti-parallel? Well, first of all, they had a little bit of evidence already. They had, from Chargaff, these results. So I want you to have a look at the results and see if you can work out how these results suggest the structure that we've already looked at. Well, let's have a look. Adenine is 31% and thymine is 31%, 31.5. Cytosine 19.1, 18.4. What conclusion can we draw? They are the same. So the percentage of adenine equals the percentage of thymine, and the percentage of guanine equals the percentage of cytosine. And so that suggests that they bond together. And so this is one piece of evidence that they used. So this was known as Chargaff's ratio. So Chargaff's ratio is the idea that A equals T and G equals C. The other thing that they had, and we'll talk about some of the history behind this as well a bit later, is they had this. And this was from Rosalind Franklin's work. And it was taken without her permission out of the lab and given to Watson and Crick. And you can see that there's this X shape in the middle. And these bits here, they are the rungs of the ladder. They are the bases. And they could see that it was kind of making this what looked like a helix structure. So they had that evidence as well. This was made through X-ray. They were shining X-rays through, it was crystallography, because they were shining it through crystals of DNA. They were crystallizing DNA, shining X-rays through and developing it. And from that, they had some idea of the structure. And this suggested a helix form. And so what they did was they used cardboard cutouts. You can see here they used clamp stands and they cut out cardboard. And they started trying to make it work. Knowing what they knew about hydrogen bonding and structure, they knew the structure of the bases themselves, but they didn't know how they fitted together. So quick summary, they used cutouts of cardboard sheeting to see how nucleotides might fit together. Initially they had an idea that it was going to be a triple helix model, but they revised it when Chargaff showed that the ratio of A to T was 1:1 and C to G was 1:1. Therefore the adenine must pair with the thymine. And in a flash of insight, they tried two anti-parallel strands, with one running one way and one running the other. So the final bit here is to have a look at why these are anti-parallel and the idea of 5' to 3'. Okay, so this 5 carbon up here, this end is called the 5'. 5'. So that little apostrophe there means 5'. That's the 5' end, and you can see if we follow it down to the end of the strand and label 1, 2, 3, then this end of the strand, this end is, it's running from the 5 at the top to the 3 at the bottom. Now if we look at the other one, and I'm going to label it here, if we look at this strand over here, well, we have to label it the same way. So 1 would be there, and this might be easier to do upside down. 2, 3, 4, 5. So we can see that at this end, it's the 3' end, and at that end, it's the 5'. So this strand, if we look down the page, is running 5' down to 3' at the bottom, whereas this one here is running, if we label this one up here as well, 1, 2, 3, that's the 3' end. This one is running 3' up here down to 5' at the bottom there. So the strands don't run the same way. This one's 5 to 3 going top to bottom, and this one is over here is 3 down to 5. I hope that kind of makes sense, and we'll go back over that. And that's it. One final repeat, sorry. Here we go. If we look at this one, that's 1, 2, 3, 4, 5. And 1, 2, 3, 4, 5. So this strand here, the bottom end is 3 and the top is 5. So this is running 5' down to the end part is 3'. If we look at this one over here, 1, 2, 3, 4, 5, and 1, 2, 3, 4, 5, we can see that this strand here is 3' with the 5' at the bottom. So this strand is running 3' to 5'.